lot to go over, so. All right, well, welcome everyone. Welcome to the Fuse Symposium and to this workshop. Certainly appreciate you coming. So I'm Mary Nilsson. So I have a master's degree from Iowa State. I've been working at Eli Lilly ever since. I'm approaching my 30-year anniversary, which is kind of scary. I've worked on quite a few submissions, either as the statistician on the project, or as a consultant, or as powerhouse. I've worked on about 8 submissions. So hopefully I can share some of the experience or things I see. So a lot of the material here, like frequently asked questions and common pitfalls, are from my experience, but also experience from the other people as part of the project team. I have been part of a safety analytics group at Lilly since 2012. So since that time, I've been in a consultant role, primarily with helping submission teams plan their analyses for safety. I'm also a co-lead of the Standard Analyses and Co-Chairing Working Group, and I lead the Analysis and Display White Paper Project team. That's who I am, but a lot of people helped with this, so certainly the co-authors and all the reviewers for the Fuse White Papers. So our project team, the Analysis and Display White Papers project team, has been around since 2012 when the collaboration started. So we have final white papers, so all those people who contributed to those white papers fed into the material for this workshop. And and the reviewers, right? All the reviewers who contributed and really helps with the discussion. Special thanks to Dee Beasley, who is our FDA liaison for the Analysis and Display White Papers project team. Wei Wing and Rebecca Revis helped with some of the slides in this presentation, and we had Lori, Jerry, Swarna, Brenda also help review these slides and help contribute to these slides in particular. So here are the pre-reads that were part of the advertisement for this workshop. So how many did their pre-reads?
Can we have partial?
All right. That's okay. We won't kick you out. But I do encourage you to read these, even if it is after this workshop. So these slides are primarily based on these 4 white papers, a little heavy on the first 3. So we have an adverse event white paper, lab vital ECGs, there's 2 of them, one that deals with the central tendency type analyses and another one that looks at the outlier shift type analyses. And then we have a demographics, disposition medications white paper. So these are the primary sources that are being used for this workshop. Another useful resource is the FDA 2010 clinical review template, if you're not familiar with that document. It is an FDA guidance intended for FDA clinical reviewers, but it is available to us to see, and we found that to be one of the most useful documents, and we reference it quite often, so that's another good thing to read. It is a big document, and to get to safety, you have to go all the way to attachment B. So we actually search for attachment B, and then that's where you kind of find a lot of the more detailed information that we found useful as source for our white papers. So as a disclaimer, this disclaimer is on all of Fuse deliverables in general, I guess. It's basically, even though Fuse does have regulatory participation, all of our deliverables, it can't really be seen as required, right? So a lot of the analyses that we have in our white papers are recommendations. They are just recommendations. We're not in the same situation as CDISC standards, right? So these are just recommendations and can't be viewed as being required by the FDA. But we, had a lot of input, a lot of discussion, so hopefully they're still very helpful. So first I'll go through a little background, go through some definitions just to help facilitate the discussions. We're on the same page with some key definitions. Then we'll talk about adverse event analyses, lab analyses, just a short, brief discussion of medications, and then give a summary. As you can see, this is being recorded. The idea is that all of you, when you go back to your companies, you can share the recording and have that be something to help with your educational goals. Right? One thing that we're trying to do is crowdsource. It's what this whole collaboration is about, is to crowdsource activities. And one of the things we're trying to crowdsource is education around analyses, right? So you can use this video to help take back to your place and use it as an educational tool.
Quick question.
Yes.
So the video going to be available on the website?
It will be publicly available, most likely, we'll have to say where it is, but if you go, hopefully when you go to Fuse EU and to working groups, it find that pretty easily. Okay.
Thank you.
Yep. All right, I will, after each section, after adverse events, after labs, we're going to have some dedicated time to deal with questions. I probably will go through some of the material without taking too many questions, unless it's urgent. Otherwise, I'll just wait till after each section just to help with the people who are viewing the video. It might just be easier to follow to have a section kind of covered at first and then with some questions. So for each of these topics, we'll discuss the recommended tables and figures, right? So these are documented in the white papers. And for this workshop, the white papers cover recommendations for both individual studies and also integrated analyses. For this workshop, we're going to just focus on individual studies just because of time. Maybe we'll have another workshop that will focus on integrated analysis, because there's a whole lot of shared learning that we need for integrated. There's a lot of common pitfalls, there's a lot of poor practices going on with integrated analyses, and we absolutely want to cover that education, but for this initial workshop, we're going to cover individual studies first. And this gives us a chance to expand on rationale, expand on interpretation. We can go over some frequently asked questions that we get not only from maybe a statistical colleague, but maybe from medical colleagues or regulatory colleagues. It goes into the frequently asked questions and common pitfalls. So why are we having this workshop? And the main reason is we have seen a gap in safety analytics knowledge. And this gap is across all functions, even in statistics. So even if you go to school, you might not learn what you need to learn to become a statistician in the clinical trial space, planning and writing and interpreting safety analyses. You don't necessarily come with the skills that you need to have. So we're trying to fill that gap, and it is cross-functional. It's a cross-functional gap. And we found in our experience is sometimes dealing with this type of education where you're in the midst of a submission or you're trying to get a study report done, sometimes it's not the time to go into a whole lot of education because it's just, you don't have enough time. You might get 2 minutes to talk about a pretty technical topic. So the idea is to set aside a little bit of time to increase your expertise on safety analyses. So it is a myth that analyzing safety is easy. You would think by now maybe it is, because we've done how many trials as an industry, and they're all pretty routine, but it really is not that easy. And a lot of study designs take a lot of thought on how to do them correctly, and interpretation may not always be easy, especially as we get into some more complex study designs. But even simple study designs, we see some common pitfalls that we need to change. We need to really elevate the expertise among ourselves and our colleagues so we can interpret our analyses better. So a little background on the Analysis and Display White Paper project. So as I said, I do lead that. Dee Beasley is our FDA liaison. She is a clinical reviewer at the FDA. And the idea here is to provide recommended tables, figures, and listings, and for both individual studies and integrated submissions. And it is to begin the process of getting to some type of standards. Right? Currently, even for just adverse events or labs that we're all doing, there's more variation that we probably need to have. Right? We still have so much variation on how teams analyze this data, and that might be unnecessary variation. And the more variation we have, think about the board clinical reviewer every time they get a submission and get these study reports. They need to learn what the company is providing, right? Or they might need to, if it's a novel plot, they might need to learn what it's doing, right? So the more we can consistently move toward consistent displays, we're going to be helping with efficiency, both maybe share some code and be more efficient how we get to the analyses, but also more efficient for the reviewers reviewing the analyses, if they can become more familiar with these types of displays, especially if we want to be more innovative with some novel figures, right? Then if we're all doing it, then the reviewers can come up to speed. So our goals on our project team is to improve expertise in safety analytics, which is the goal that we're dealing with today, right? Having a workshop and going through some of the statistical thinking involved in planning safety analyses. So we're trying to promote good analytical practice and start trying to avoid more of the poor practices. So define a set of recommended tables, figures, listings associated with the analyses. The idea is if we can try to make sure the reviewer receives relevant tables and figures, and also harmonization leads to efficiency, so people start getting used to the same figures. And the idea is to facilitate cross-functional engagement in the analytical planning. So we have been around since 2012. We do have 6 white papers that have already been finalized, and these are all publicly available. You go to fuse.eu and click working groups with white papers. You'll see all these white papers, not only from our project team, but the entire Fuse collaboration. All the final deliverables are in the deliverables catalog. So the ones we'll talk about today, average mets, vitals, labs, demographics, medications. There are the other 2 we have final that we won't be talking about today are non-compartmental PK, QT, QTC studies. All right, so let's get into some definitions to facilitate what we're going to be talking about today. I'll say signal detection a lot, and I know that for most people, signal detection is something to think about when you have a drug that's being used in the real world, and you get data back, and how do you look at that data to get a signal detection? Well, I'm still using it even in the clinical trial space, just for convenience. If someone has a better word for it, happy to hear it, but we're using that just to mean it's the purpose. If the purpose is signal detection, it would be analysis of displays used to determine which events or changes in labs or vitals require further evaluation or further scrutiny. It deserves some attention, right, for determining whether or not you think it's an adverse drug reaction or reasonably likely caused by the drug. Signal clarification is a word we use to say, well, these could be additional analyses and displays you may need to facilitate your final assessment whether you think the event or change is reasonably likely caused by the drug. Signal characterization is once you determine something is likely caused by the drug or is an adverse drug reaction, more often than not, you want to say something about that event. You want to say, does it occur early? Does it occur late? Is it persistent? Is it transient? We use the words signal characterization for those type of analyses. And signal communication would also include analyses you might want for the main purpose of communicating an event. So what you would put in a drug's label, right? So you might want to put something in the label, and it might need to match what competitors have, so it might be something extra you need, or you may not need any extra things for this, but you might have some summaries that have the primary purpose of communicating that adverse event. Okay, another thing that we'll talk about is the difference between a static display and an interactive display. So a static display is a table, figure, or listing that you can create that just basically can sit on a paper, or it could be digitized paper, right? I mean, it could still be electronic, but it's going to fit on a page, right? So that's a static display. Okay, interactive display is something that has some type of point-and-click technology associated with it. So you might be able to zoom on something, or hover over something, or lasso over a point and see extra data, right? So an interactive display is something that has some type of interactive feature with it. Okay? The other thing that is just some advice or convention when we talk is that, and I hope I don't make this mistake, but we try to be very careful to say, you know, if we say the FDA thinks this, or the FDA says this, or they believe it, we like to reserve that kind of language for when there's an established guideline or an FDA guidance. Otherwise, we've seen this habit even at the Fuse collaboration, and some of our FDA colleagues will catch us and say, well, the FDA said that. It's like, well, who said that? It's like, well, somebody in one of our meetings. It's like, well, no, that's not the FDA. That's just the person who happened to be participating at that meeting, right? So we have a tendency, if someone from a regulatory agency says it, we have this belief that that must be what the whole agency thinks, right? And that's not true. So we are trying to be careful with this, and I would encourage you to always be careful of this here throughout the symposium, but also when you go back to your companies, to just be careful, to not overgeneralize, because the FDA regulatory reviewers are their own people and have their own opinions. So we do try to be careful. So this is one of the first common pitfalls we're come to, is it's really easy to assume that if you had an interaction with the regulator with one of your drugs, to assume that the next clinical reviewer might have the same opinion, and that's not something that we have seen. We've seen definitely a lot of different opinions across the agency, and of course across within our companies too. You can't take one person's opinion and all of a sudden, well, that's that company's opinion, right? So we do need to be careful with those language. So let's talk about what an adverse drug reaction is, because a lot of our recommended analyses in our white papers, sometimes we say the run-of-the-mill safety, right? The kind of safety analyses we always run on adverse events and labs and vitals. The main goal is signal detection. And why do you do signal detection? You're trying to figure out what you would call reasonably likely cause for your drug, or an adverse drug reaction, right? So an adverse drug reaction is an event or change in lab or vital or something that you have reason to believe is caused by the drug. Okay, then how do you determine it? It's a common pitfall, so this is our second common pitfall, that there's this belief that this decision on what's an adverse drug reaction of a drug is based on some numeric criteria, some statistical criteria, right? 1% with drug and greater than placebo, then it's an ADR. That's not true. I think most people would know that, but it still occasionally comes up where there's this belief. So we are trying to educate that is not all there's to it for an ADR to be an ADR. It's actually a whole lot of factors. Of course, how strong your evidence is for an imbalance, right? The more imbalance you have between your drug and placebo, that's going to be, more indicative of being an ADR. How you're strong, the magnitude effect takes into account any kind of dose response matters, right? So, but there's also biological plausibility. There's clinical relevance in any individual case. You could have one case for one event that's just so convincing that that event can be an ADR. The severity, the consistency of the findings across studies, how consistent they are with similar events, and then consistency across similar compounds. So what happens in another compound absolutely matters on what, if it's in the same class, it absolutely matters on your decision making on whether or not you think your drug is an ADR. Now what's in red are the factors that we can help as statisticians, right? So some of these are very much medical judgment, but there's a lot we can do within statistics to help cover a lot of these factors. And so certainly we're looking for imbalances, we're looking for magnitude, which is why you'll see in our recommended analyses, you'll see comparisons with placebo. We do advocate for risk difference or odds ratio that not everybody does, right? So we advocate for having an estimate of magnitude of effect. We certainly have tables looking at severity. So this is the basis for a lot of the recommendations we have in our white papers. Okay, so the purpose of these initial sets is signal detection, right? So it's really easy. I see this a lot for teams who have special topics in mind, right? Say, well, I'm interested in this special topic, and then if I want to know what I need to know for that, then we need these analyses, right? And they try to apply that to all of safety, right? So they might think, well, because I need this for, let's say you're interested in infections. Someone might say, well, I need this and this and this and this to look at infections. Well, that doesn't mean you have to do that same approach for every adverse event and every lab or vital, right? So that's a common pitfall. People try to bring that forward to the run-of-the-mill safety, and all of a sudden your volume of analyses just explodes, right? And then you have a lot of tables and figures that are probably unnecessary. So we also see as a common pitfall is to a priori plan for everything that covers signal detection, clarification, characterization, and communication right off the start. Well, most of those you don't need for every adverse event. You just need it for events that turn out to be adverse drug reactions for purposes of your labeling, right? But that doesn't mean you need that whole pile of analyses and apply it to every single event. And that is something we sometimes see. Also, occasionally, I've had one interaction where, all the competitors had 12-week data. Safety was 12-week data. Well, our studies were 24-week data, right? So they're saying, well, we have to do all the safety analysis with 12-week data because we need that for the label. It's like, well, no, you might need to do your, if you need that for your label, sure, do what you need for the label, 12-week data, but that doesn't mean you need to apply that for all of safety, all your run-of-the-mill safety, because you don't need that to sneak into signal detection. So that's why we're trying to think through detection, communication, clarification, to help think through what you actually need and try to right-size volume, because it's not in anybody's best interest to have a bunch of tables and figures that don't serve any purpose. All right, so let's bring up this study design, and we'll talk about this design in a little bit more detail. So this design has a screening period, has a double-blind treatment period, followed by an open-label extension period, and then a post-treatment off-drug period. So we'll have this study design in mind as we talk through a few issues.